All right, welcome everyone, and thank you for joining us for today's webinar titled Getting Started with In Vitro Blood Vessel Research. My name is Andy Henton, and I will be your host for today's event. Our session today is sponsored by Living Systems Instrumentation and is an introduction to the essential tools and techniques that scientists need to know for studying function in isolated blood vessels using a pressurized arteriograph system. First, we will hear from Dr. Gerry Herrera, President of Catamount Research and Development and Living Systems Instrumentation. Catamount R&D is a laboratory services company specializing in preclinical research studies, and Living Systems Instrumentation designs and manufactures instruments for in vitro research studies of perfused cannulated blood vessels. Today, Gerry will be providing a brief historical overview regarding the study of isolated vessels and discussing typical system design and laboratory setup for pressurized arteriographs. Following, we will be joined by Dr. Scott Earley, Associate Professor of Pharmacology at the University of Nevada School of Medicine. Scott has been studying vessels using these techniques for over 15 years and is going to cover the essential steps for getting started with these experiments, including tips and tricks that he has learned along the way that will ensure consistent and accurate results.
Okay, thank you, Andy. And I want to thank all of you for attending as well. We're really happy to have you all from all over the world joining us today. And real excited about today's event, and I hope it's useful for you all and that you're able to get some good information and resources out of today's meeting. Let me just start by telling you a little bit about my background, how I got to where I'm at and what we do at my companies. Again, my name's Gerry Herrera. I'm the president of Catamount Research and Development and Living Systems Instrumentation. My background is in biomedical research. I actually, going back to as an undergraduate student, I studied cardiovascular physiology in Ben Walker's lab at the University of New Mexico. I think we have a few people from New Mexico joining us today, so welcome. And after leaving Ben's lab, I wanted to learn more in vitro techniques for studying the cardiovascular physiology. So I actually came to the University of Vermont to do my PhD with Dr. Mark Nelson, and I did my PhD in molecular physiology and biophysics. I actually shifted gears a little bit and studied smooth muscle excitation contraction coupling in urinary bladder smooth muscle. But I've always been very interested in cardiovascular physiology, and it was actually while I was at the University of Vermont that I became friends with someone that many of you attending may know, Dr. Bill Halpern. He was a professor of physiology at the University of Vermont and is one of the pioneers, really, of in vitro studies in the cardiovascular system. And it was really a great pleasure in my life to be able to know Bill for a few years. Bill passed away in 2008. And as many of you know, he founded the company Living System Instrumentation, and where he would innovate the instruments that he designed in the lab and actually made them into systems that other people in the research community could use. So it was actually, after I left my PhD work and finished, I did a short postdoc, I actually shifted gears and left in academia and started working with a company called Med Associates. And I really enjoyed this side of science where I was working with engineers, electrical, software, and mechanical engineers, and helping to design and develop research instrumentation and bring them into the lab for practical use. And that led to me deciding to start up a new company, Catamount Research and Development, where that was our mission to develop and refine biomedical research instruments. And so I really brought a basic science approach to product development at Catamount Research and Development. We actually have an in-house laboratory, animal care facility, institutional animal care and use committee. We're set up to do basic science here, and we use that in our product development approach. So it's a very unique approach in the industry. Not a lot of other companies have that kind of approach to developing instrumentation, where we really take things through from basic science all the way through development and end up with products that go out into the lab. So we're real excited about what we do here. And it was through this course that I maintained contact with Bill Halpern at Living Systems Instrumentation, and continued to be in contact with him over the years. And to see if there's ways that we could work together with our two companies. And it was actually when he realized he was diagnosed with pancreatic cancer, he decided he wanted to see his company go forward and continue. And so I was really happy to have an opportunity to purchase his company shortly before his death. It was really sad to lose Bill, a mentor and friend, but I was really honored and excited to have an opportunity to keep his company and legacy going forward. And that's what I wanted to talk just briefly about, was how we got where we are with cardiovascular instrumentation. Here's a picture of Bill. And Dr. Halpern and his colleague Michael Mulvaney did a study together back time. In vitro studies in vascular tissue were limited primarily to large vessels, conduit arteries, and so not a lot of functional information was known from direct studies of tissue from resistance arteries, those blood vessels in the body that are important for controlling vascular tone and vascular resistance. So Dr. Halpern and Mulvaney worked together to try to address that, and they described a project that was published in Nature in 1976, where they recorded the first time measurements of mechanical properties of vascular function from resistance blood vessels. And here's a schematic of the instrumentation that they developed at that time. It's become known as the wire myograph, where they took a small isolated segment of a resistance vessel and mounted it between two support structures. And the vessel segment was held in place by two wires passed through the lumen of the vessel. And so one of the support structures was maintained fixed, while the other support structure could be attached to a force transducer. And this allowed them to measure directly the contractile forces generated by this tissue derived from a resistance artery. And this is a figure from their Nature paper. This is important because it's the first recordings first functional recordings of vascular function from resistance blood vessels. It's a length tension curve. So we're looking at tension change as a function of length perturbations. And so these are the very first recordings of functional measurements from resistance blood vessels. And this wire myograph became a very important tool that's continued to be used today all over the world for important studies in vascular function. In Dr. Halpern's lab, he continued to seek a way to study blood vessels closer and closer to their physiological state. So he wanted to take the blood vessel and be able to pressurize it and study it in an isolated manner in which he could control intraluminal pressure and record changes in arterial diameter at various intravascular pressures. And that was that led to the development of the first arteriograph for in vitro vessel research, which Dr. Halpern's group published in the early 80s. And here's a figure from his initial paper showing a schematic of the first pressure arteriograph. And this is the vessel chamber in which the blood vessel is mounted between a glass pipette and held closed at the other end. So one end of the vessel is attached to a glass pipette, which you could control with a pressure source, and then the other end of the vessel would be clamped shut. At this time, the arterial diameter was measured visually and optically just by looking through an ocular on a microscope, which had a dimensional reticle inside of it. So they would manually take measurements of the arterial diameter and record those observations in their notebook. And this approach was really important. Over here in this figure from the Earley paper, we see the first recordings of a myogenic response from a rat cerebral artery. And in the lower curve here, this is the recording from a blood vessel that's been constricted with high potassium solution to depolarize and maximally constrict the vessel. And then the upper curves show we have a passive diameter curve with the circles. So this recording was obtained from a saline solution lacking calcium, in which we just see the vessel's passive response to increasing transmural pressure. We see a passive dilation. And then in a normal physiological saline solution, we see the tracing with the triangles here, where the vessel constricts in response to increasing in arterial pressure. And that response where the vessel constricts in the face of increasing pressure has been termed the myogenic response. It's so important for in vivo function and regulating blood flow in the face of changes in blood pressure. So this recording with an arteriograph was the first time that this myogenic response could be directly measured in a resistance tissue. Dr. Halpern continued to design and develop instrumentation to help aid their measurements. One of the innovations was automating data collection using an edge detection system. And this was published in the mid-80s where they took their pressure arteriograph, and instead of measuring the diameter manually, they attached a TV camera to their microscope and then sent the TV signal into a video dimension analyzing system, which has a processing circuit that allowed them to measure the wall thickness and intraluminal diameter based on the changes in the contrast of the video signal. So we see a dilated blood vessel here and the same blood vessel that's been constricted, changing arterial diameter, the changing wall thickness. All of these things can be tracked with the video dimension analyzing system. And this just shows a video clip. This is actually one of Dr. Halpern's recordings from his lab from a rat mesenteric artery that's constricted with high potassium. And you can see the dimension tracking at work here where as the diameter constricts, the wall and lumen is tracked with the tracking system. So a real important device that helps in vitro cardiovascular studies. Now, I want to spend a couple minutes giving you an overview of the equipment that you need to set up for isolated in vitro cardiovascular studies. And Dr. Earley, who will be joining us here shortly, he's going to spend some more time talking about a lot of these components. But this is a schematic that you might find useful if you're looking to set up a system in the lab or add on a capability is in the lab. You can kind of use this schematic as a guide of what are the key points you need to consider. And this is a basic arteriograph system that's set up for controlling intravascular pressure. And the vast majority of applications are well served by this type of a system that allows you to control the intravascular pressure in which there's no intraluminal flow. So in this case, here's the vessel chamber, which would be sitting on a microscope, and the distal end of the vessel would be closed off with a stopcock or simply tied shut. So there would be no intraluminal flow in this application. The proximal end of the vessel is attached to your pressure transducer and a pressure control system. Now we have an automated pressure servo controller that you can dial in your pressure set points, but there's many ways of doing this. You can, many labs simply hang a reservoir of saline from the ceiling and adjust the height of the water column to regulate intravascular pressure. And there's other ways of attaching pumps and pressure controls to maintain intravascular pressure, but this is the basic concept. So other key components would be a means for controlling the temperature of the preparation to maintain physiological temperature. Most of the resistance vessels are very sensitive to temperature, and vascular tone will change as a function of temperature. So temperature control is an important aspect. And then, of course, the data acquisition hardware and software is also an important consideration, whether that's done with a video camera or done with computer software. You have to have some way of capturing and storing the data. And so this is your basic setup for controlling intravascular pressure in an isolated blood vessel, which the desire is to control intraluminal flow as well as intravascular pressure. We're not going to be talking too much about intravascular flow today, but I just wanted to talk briefly about it here just to introduce this as a possibility where the desire is to control intravascular pressure and also have a constant or variable intraluminal flow rate. And this allows you to study things like flow-dependent dilations or flow-dependent constrictions or other responses mediated by shear stress. And so this is a schematic of the components that would be considered for setting up a system for doing constant pressure and flow, intraluminal flow control. So it's a little bit more complex. In this setup, you have the vessel chamber, and instead of closing off the distal end of the vessel, the distal end would remain open into a perfusion circuit. And you have to have a pressure transducer on the downstream end of the vessel. in addition to a pressure transducer on the upstream side of the vessel. So you have two pressure transducers. We're recording upstream and downstream pressure. And then the pressure control instrument would be regulating based on the average of those two pressures. So you're controlling the intravascular pressure. And that's an important aspect of the intraluminal flow setup. In addition to the pressure control, we also need a means for controlling flow rate. And that can be done with a peristaltic pump, a syringe pump, or there are other styles of pumps that can be used to generate intravascular flow in this type of a setup. The rest of the equipment remains similar to the basic setup. You need temperature control. You also need data acquisition software and hardware, and that's all outlined here. So that's an overview. Hopefully these schematics will be useful to you as a resource. You can download the slides and have them available offline and have these schematics to refer to later. So finally, I just want to talk a few moments about the parts of a pressure arteriograph. This is a current generation arteriograph that we make here at Living Systems, and it shows the different parts of the system. So this would be looking down on the vessel chamber here, sitting on the stage of a microscope. Most times an inverted microscope would be used, and so there's an optical cover slip that allows the objective beneath the chamber to image the blood vessel. And the blood vessel would be mounted between these glass pipettes here. And here's a micrograph showing a blood vessel mounted on two glass pipettes. You can see how the vessel is held in place by nylon threads here on either side. And then the chamber also allows for superfusion. So there's a fluid path that allows you to superfuse your physiological buffer into the bath and pass it out the other side. And so you'll also have the ability to measure temperature and control the temperature in the bath to keep it as a physiological temperature. And another important design feature of the arteriograph are the micromanipulators here that allow you to position and align the glass pipettes on either side. And so I'm really happy to have Dr. Scott Earley here today. I consider Scott a good friend and colleague. We've overlapped and had a sort of a similar academic and educational path over the years. I've really enjoyed working with Scott, corresponding with him. Scott recently took a new position as an associate professor of pharmacology at the University of Nevada in Reno. And so I'm real happy to have Scott here today, taking time out of his schedule. You know, he's setting up his new lab and getting his new lab up and running, so I really appreciate him being here today. Scott's really an outstanding investigator. He does leading research in the field of local control of vascular function. He's done important studies on the role of trip channels in vascular tone, local calcium signals that are important in endothelium-dependent hyperpolarizations. Most recently, Scott's group published a paper in Science Signaling using a multifaceted approach, including calcium imaging records of calcium entry through single TRPA1 channels to demonstrate a role for lipid peroxidation metabolites in triggering calcium entry through TRPA1 channels in endothelium-dependent control of our cerebral artery function. And this was put forth as a mechanism for endothelium-dependent dilations stimulated by reactive oxygen species. So really some important work. Scott, are you with us here?
Yes, I'm here, Jerry.
Okay, thanks. Again, I appreciate you being here, and
all right.
Over to you.
So I just want to thank Jerry and Andy for their efforts and for inviting me here today. And it's a real pleasure to be here working with Jerry. He's been a great friend over the years. And my purpose in being here today is to kind of present you with the workflow from the perspective for this technique, from the perspective of somebody who uses this in their lab. And I use this in my lab on a daily basis. You know, on any given day, we do isolated vessel experiments. I have 3 experimental rigs for for doing this, and and I have at least 1 or 2 postdocs every day is pretty much engaged in using this technique. So what I'm hoping to accomplish is to present you with what your workflow might be. I'm not going to be able to answer every single question you might have about performing this technique if you're an absolute beginner, but I can at least I hope to put you off to a good start. So in terms of the, I guess it's first, the first thing to consider is what kinds of studies can we do using pressurized arteriograph systems. And so we know blood vessels are composed of smooth muscle cells, which kind of make up the meat of the wall. And these are contractile cells that are involved in the control of the luminal diameter of the vessels. And so then the lumen of the blood vessels is lined with a single layer of endothelial cells. And you can think of this kind of like a Teflon coating of the blood vessels. And so the endothelial cells are involved, of course, in they have a number of factors, but in terms of vascular reactivity, they're involved in producing substances that regulate the contractility of smooth muscle cells. And most prominently, they're involved in relaxation of the smooth muscle cells. So using the pressurized arteriograph systems, we can investigate the function of these two types of cells and how these two types of cells work together to control vascular diameter. So that's what my presentation is going to focus on today. So now I know many of you, based on the audience polling, are really interested in wall thickness and vascular remodeling. So we can use the arteriograph systems that we talk about to actually measure the changes in the wall and to measure vascular remodeling. I'm not going to talk about those techniques today, but I mean that's certainly something that many of you have interest in. Okay. So what are we going to cover? So number one, we're going to cover basic equipment and procedures. So, Gerry kind of showed you schematically, he showed you the theory, and I'm going to show you the practice. I'm going to show you, what this stuff actually looks like in a working lab. So then we're going to go through some example experiments. And these are kind of the steps that people that come into my lab, this is how I train them in order to become expert on this technique. We're going to talk about potassium chloride and phenylephrineinduced constriction of mouse mesenteric arteries. Mesenteric arteries are the easiest arteries to work with, and so I would suggest if you're getting started with this technique, you start there. So by looking at KCL and phenylephrine-induced constriction, we can look at smooth muscle cell function. Then we'll look at acetylcholine-induced dilation of the mesenteric arteries. This is a way of looking at endothelial cell function. And finally, at the end, I'm just going to show one example of myogenic constriction. And I'm going to show you, this is sort of maybe one of the limits of this technique. And so we're going to talk about mouse cerebral parenchymal arterioles. So these are the small arterioles that bridge the peel arteries on the surface of the brain with the subsurface microvasculature. And so these are critically important for vascular function in the brain, and that it's a current area of interest in my lab. And finally, after we show these example experiments, we're going to talk about, we're just going to go over a few tips and some troubleshooting things that you can do. Okay, so how to get started. So the first step, if you're coming in, you're starting your experimental day, the first thing you need to do is prepare the solutions. So this just shows you Harry Pritchard. He's a new postdoc in the lab, and he's hard at work doing this. And so for the purposes of the experiments that we're going to talk about today, you'll need to prepare 4 solutions. And so the first solution will be this MOPS buffered saline. So this is a biologically buffered saline solution. And we use this solution for isolation of the tissues. So we'll isolate the organ, say the mesentery or the brain. We'll isolate the organ into this solution. We'll maintain the tissue in this solution until we're ready to do an experiment. And this keeps the solution stable. This keeps the tissue stable and happy during the procedures. So we'll make this and we'll put this on ice. We'll keep it cold. The next solution you need is what we call PSS, or physiological saline solution. This is a bicarbonate buffered solution, and this is you make maybe a couple liters of this, and this is what we use to superfuse the vessel. We use this to perfuse the vessel. This is the main solution that you'll use throughout the day. We'll also make a high potassium PSS. And so this is this basically we isotonically substitute potassium for sodium chloride in the physiological saline solution. So we try to make a 60 millimolar. We make a 60 millimolar extracellular potassium solution, and we use this for looking at KCL-induced constriction of the blood vessels. And finally, you'll need a calcium-free PSS. So this is exactly the same composition as the main PSS solution, except for we haven't added calcium, and then we've added a calcium chelator called EGTA to remove any residual calcium. And we use this solution in order to establish the passive responses of the blood vessels. These are the way the blood vessels respond if we remove the ability of the smooth muscle cells to constrict. The constriction of smooth muscle cells is calcium dependent. If we remove calcium from the extracellular solution, the vessels can't constrict and they behave basically like passive tubes. So we prepare all these solutions. And one thing that I want to emphasize is, if you want to be successful at this technique, is that you prepare all the solutions on the day of the experiment. So we find that in our hands that if we try to save these solutions overnight, we get stuff precipitating, we get stuff growing, and we don't get the results that we like to have. So in my mind, I always tell the postdocs on a daily basis, just make your solutions fresh every day. It only takes a few minutes, and it will save you lots of trouble down the line. So there are other, and you can get the composition of these solutions from my publications. There are other ways. Sometimes people like to use Heppy's buffered solutions for these procedures, and these work well in many people's hands. The advantage is that you don't have to bubble the solutions for the physiological saline solutions, because they're bicarbonate based. You need to bubble with a CO2 gas mixture in order to get the appropriate pH. If you don't want to do that, you can use these Heppy's buffered solutions, and I believe that the compositions of those Heppy solutions will be provided to you with the materials from this webinar. So first step is to make the solutions. The second step is to isolate the blood vessels. So what you need in order to isolate the blood vessels is you need a good dissecting microscope, shown here on the top panel. I don't have any brand loyalties. I don't know that any brand of microscope is better than any other brand. We have two of these microscopes in the lab. One is an Olympus and one is a Nikon. The one thing I do like, though, is I do like to set up my dissection microscopes on the boom stand. So you have a nice heavy plate here, so that makes the microscope very stable, and that really helps me out a lot when I'm doing the fine dissection and when I'm trying to cannulate the blood vessels. So it really doesn't matter, any particular configurations, whatever you're comfortable with, whatever works, and as long as you have a nice stable platform and you have enough working distance to get your tools to where you need to have them. You also need to have some dissection tools. So you need some fine-tip forceps. So we have we like to use these number 5 forceps. I like to use the titanium ones. Some people like to use stainless steel. Matter of personal preference. Really important to keep them nice and sharp, not ding them around. Keep the points as fine as possible. And then you also need a set of small scissors. These are sometimes called Vanna scissors or microvessel scissors. So, you can obtain these from various vendors. You can get them from Small Instruments Incorporated or various vendors. So once you have your tools, then you need some dissection dishes. And these are the dishes in which we pin out the tissues from which we isolate the blood vessels. So we use 2 sizes in the lab. We have these shallow ones that are shown here, and then we have deeper ones shown here. The shallow ones we use for isolation of mesenteric arteries, and I'll show you a picture of that here in a minute. And then the deeper ones we show for isolating for isolating cerebral arteries. So we'll put the brain in here. You can buy these from various vendors, or you can make them yourselves. We make them ourselves. So we'll buy Silgard, and we'll buy glass Petri dishes, and then we just pour the Silgard to the depth that we want. And you can see that these are, these see a lot of use. You can see how the Silgard is getting pretty beat up here. You also need a set of insect pins. So these are the pins that people who collect butterflies or whatnot use to pin down their insects. You can buy these from various vendors, so you'll need a set of those. In my case, I bought them once, and I basically have a lifetime supply. Sometimes we use smaller dishes for cleaning up the arteries, and you can see one of these here, and this is just a small 35 millimeter Petri dish in which we poured Silgard. So once you've assembled all of that stuff, you can sacrifice your animals. Of course, you need to follow your IACOP recommendation for humanely euthanizing the animals and for collection of the tissue, and you need to deal with all of that regulation up front. After you isolate the tissues, you can pin them out in these dishes. So this just shows you two examples. So this shows a mesentery. So this is a mesentery pinned out in a dish. So you can see how we use the insect pins to kind of stretch the tissue out just a little bit. You don't want to stretch it too much. You don't want to damage the arteries. But you want to be able to visualize the blood vessels so you can isolate them. So you can kind of see an example like this. So it's almost like these things look like the spokes of a wheel. And so you can get in and you can isolate the tissue that way. This shows you the example of a brain is pinned out. So this is a mouse brain. This is the distal surface of the brain pinned out. So you're looking down on it, kind of looking down on it. This is the circle of Willis. And so you can isolate the peel arteries that come off the circle of Willis. You can quite clearly see the middle cerebral arteries coming off the circle. Here, this would be like this, and then from here on out, we're going to focus on the mesenteric arteries. And so you can see here, this is the same mesenteric prep that we were looking at earlier, but this is with the fat removed. So you need to go in and you sort of blunt dissect away all this fat. You can see all this fat that's coating the arteries up here. So you blunt dissect this stuff away. Obviously, you want to be gentle. You don't want to damage the blood vessels, but you do want to get them nice and clean. So you can see. You can see how these arteries are all coated with fat, but then you can see how we've cleaned away all the fat in these. So then the big thing you want to do here is that you need to distinguish between the arteries and the veins, right? So we want to study, most of the time we want to study arteries, although you may want to study veins as well, but you need to know which is which. And then this prep is quite easy to see. You can see the arteries are the very thin vessels, the smaller vessels. So you can see how small these are. They don't have a lot of blood in them. Whereas the arteries are these nice big fat, kind of flaccid vessels that are full of blood. So after you get the fat cleaned away, then you can identify the arteries and the veins, and then you can cut these out. So you use your dissection scissors, make a couple cuts, and pull these out, and then store these. And we use small vials. Just small glass vials to store these until we're ready to do an experiment. And so on any given day, you might come in and you might isolate, several of these, quite a few of these, maybe 6 or 8 of these, just to have them ready to go. Keep them in your little vial with some multi-buffered saline or whatever you're storing your tissue in, and just keep them. Hold them on ice, and then you have to mount them in the pressure myograph chamber. And so where you can mount the vessel ties. And the way we do this in the lab is we have these braided nylon fibers. You can buy these from various vendors. You unravel a piece of that, maybe 5 to 10 millimeters long, maybe longer. Doesn't really matter at this point. And then you want to unravel these. So these things are composed of multiple filaments. So you want to unravel them until you have an individual filament. So then you use one filament per tie. So you take your filament. I do this under the microscope. I know some people that have good enough vision and dexterity to do this without a microscope, but I think most of us have to do this under the microscope. But you simply make a very simple half hitch. This is called a half hitch. It kind of looks like a pretzel here in this picture. So you make a nice little half hitch using your fine tip forceps, and then you can stick these down to, usually what we do is we put some two-sided tape in a petri dish and we stick these down. And I actually use the tape to actually make the tie. I find it convenient to have the string kind of stuck down a little bit. It's just a little bit easier for me to handle. It takes a little bit of practice. So this is maybe something you do before you actually get ready to do an experiment. Maybe spend a few afternoons here and there just kind of practicing making ties. It's really good for getting your hands trained for doing this fine dissection work. So you make a bunch of these ties. You'll need at least 4 per experiment. Good idea to make extras. So maybe sit there and make 10, 12, or 20 of these or so, and just have them all stuck down in your dish and ready to go. So after you make your ties, the next step is actually mounting the vessels in the pressure myograph. And in many ways, this is kind of the rate limiting step, I think, for a lot of us in terms of doing these experiments. It's the most intensive kind of hands-on technique. It takes a little bit of manual dexterity. It takes a little while to get a feel for doing this. So if you've got good steady hands, you should be able to do this without much problem. But essentially, what you do is you take a vessel segment as shown here, and you try to find a segment that's free of any side branches or anything, any fat or anything like that. So a nice clean vessel segment that's sufficiently long to work with. And you take that vessel segment, you put it in the pressure myograph chamber, and you slide the end of it onto one of your glass cannulas. So you can see the glass cannula here. So again, you use your fine tip forceps. You slide the vessel over the end of the glass cannula, and you just basically pull it on like a sock. It's really you want to get enough cannula in the vessels so that you have something to tie onto, but you also want to leave enough vessel free so it can actually constrict, and so it can actually do its thing. So you get the vessel pulled on, you put a tie on the end of it. So you take one of your ties, you kind of maybe I usually put the ties on the cannula before I start the experiment, so they're already in place. Slide it down the vessel and then cinch it off, and you can tie it off. So you can see how it's done here. Many labs use 2 ties. This kind of gives you a backup in case you have a leak or something like that. This may help you out if you're just getting started. You may want to use 2 ties. Once you get the proximal end cannulated and attached, you want to just put a little tiny bit of pressure on this, and you can use a syringe for this. But just don't just cram down on it and hit it too hard. You just want to use the most gentle pressure you can that will actually flush the blood out of the lumen. You don't want to have any blood in the lumen here, so you put a little bit of pressure on here. You flush the blood out. Once you've done that, then it's time to cannulate the distal end. So you maneuver the vessel in place using the controls on the vessel chamber. So you get this distal cannula positioned so that you can pull the vessel over the end, and then you tie it off. Okay, so once you get that done, then you want to then the next step is to transfer this chamber to your recording system. So then you take the vessel chamber out from under your dissection microscope, and you put it on your recording system. So usually, again, like Gerry said, it's an inverted microscope, so you can see what we got going on here. You can see the vessel's cannulated here, and the microscope objective is here. You connect the inlet and the outlet superfusion systems. So you connect the superfusion tubing. So you can see that there's an inlet coming here, and then an outlet coming here. So you connect these up, and then you connect your pressure control system. So you can see this is what we have here. So we've got one end of the vessel tied off here, and so then we connected our pressure control system through this port here. So you get it all set up here, and so then your next job is to keep the vessels alive and happy. So obviously we want these vessels to be able to constrict and do their thing, and so the parameters that you want to think about controlling at this point are temperature, flow rate, pH, and intraluminal pressure. So in terms of temperature, we do our experiments at physiologic temperature. We around 37 degrees C. You can see for this experiment, this is a temperature monitor. So sometimes you can put a thermocouple in the bath and record the temperature if you want. You can see this experiment was being performed at 35 degrees C. In order to warm the superfusion solution to 35 degrees C, we use these Warner temperature controllers. These have these nice online cartridge systems. You can also do a counter current approach using a heated water bath. We use the cartridge heaters basically because they're quieter. It just keeps the lab a little bit of white noise out of the lab. So you can see in this case we had to warm the solution up to about 48 degrees in order for it to be delivered to the chamber at close to 37 degrees. There's also we can also, as an auxiliary system, we can use a temperature control that actually heats the bath itself. So we use both approaches. So we'll warm the superfusion solution to achieve 37 degrees, and then we kind of do fine control using these temperature control systems that attach to the bath. In terms of a flow rate, so we're superfusing the vessel. We've talked about that a little bit. And so we superfuse the vessel. We use these little Masterflex pumps. So you'll need a Masterflex pump pumping the solution in. We use a flow rate of about 3 to 5 mils per minute. That's pretty typical. I'm not actually sure where those values came from. They've just been in use probably since Bill Halpern's day, and so we've just continued with those practice. So keep in mind that if you're pumping fluid in, you've got to remove fluid from the bath. You can do that if your lab is equipped with house vacuum. You can attach a vacuum flask to the end of your chamber on the outflow side and remove it that way. Or if you don't have access to house vacuum, you can use another pump. Just keep in mind that the flow rate, you want to make sure that the flow rate coming out is greater than the flow rate going in. One of the worst disasters you can have is that if you overflow your chamber and you get all that salt solution down into your microscope, then you might have to pay somebody to come out and repair and clean your microscope, and we like to avoid that. It's important to maintain physiological pH if you want to keep your vessels happy. We do this. We use a bicarbonate buffered solution. So we bubble the solution with a CO2 containing gas mixture. In my lab, we use a gas mixture that's 6% CO2, 21% oxygen, and the balance is nitrogen. So we use 6% CO2 because we're here in Reno, we're at about 5,000 feet. If you were closer to sea level, in order to maintain physiological pH of 7.4, you might want to use a gas mixture that's closer to 5% CO2. But we just bubble at a certain rate. We use these stainless steel diffusers in order to just make sure that these bicarb buffers stay at a pH of about 7.4. If you're having trouble with contractility, especially if you get a lot of basal motion artifacts in your blood vessels, it's probably a pH issue. And so you'll want to monitor the pH. You'll want to ensure that the pH is correct. But actually, your blood vessel is actually your best pH sensor that you can have in the system, because if the vessel is behaving properly, your pH is appropriate. So finally, we need to control intraluminal pressure. And the way we do this in my lab is that we use these servo pressure controllers. So these are very nice because you can dial in whatever level of intraluminal pressure you have. There's a pressure monitor. There's a peristaltic pump. You can kind of see the corner of it up here, and it will basically increase. It will basically pump forward or backward in order to match the pressure setting that you set here. So the final step that you want to think about at this point is that you don't want your blood vessels to be leaking. You want to make sure that there are no side branches, there's no gaps between your ties and the cannulas or things like that. So you want to check your vessel for leaks. And so the way we do this is that we turn the servo controller, we turn it from automatic mode. You can see it's on automatic mode here. We'll turn this to manual mode. So if it's on manual mode, this simply becomes this simply will just read out the pressure. This takes the pump out of the circuit. So so then if this pressure begins to drop, then you know that you've got a leak. So if you see that the pressure is starting to drop when you're in manual mode, you've got a leak. So that means what you have to do is you have to take your vessel down off the recording microscope, go in and try to see if you can see where maybe there's a side branch or something, see where it's leaking, see if you can get rid of the leak. If you can't, then you're kind of stuck with taking that vessel down and putting up another blood vessel. Okay, so this is what you need to keep the vessels alive and happy. The next step, of course, is collecting the data. Gerry talked to you about the edge detection system, and so this is just kind of how it looks in my lab. And then you need a data acquisition system. You can use whatever you want. I mean, these edge detection, the output is pretty easy to calibrate. We use right here we're using a software called iWorks. And it's quite compact. It fits on, a single bench, and so it's kind of nice that you can cram several of these into your lab if you want in quite a small space. So now we'll go through some example experiments. And so the first experiment that I want to talk to you about is potassium chloride-induced vasoconstriction. And so basically what we do here is that we expose the vessels to elevated extracellular potassium. And so in this case, we'll increase extracellular potassium from 5 millimolar, which is physiological, to 60 millimolar. And to do this, we'll superfuse the vessel with our high potassium PSS. And so what we do is that we'll have a reservoir where the pump is pumping out PSS across and superfusing the vessel. So we'll just pull the tube out of that reservoir and put it into a second reservoir that contains the high potassium PSS. And so one thing that's really useful to do at this point is to keep that outflow tube out for a little bit and introduce a small air bubble in the line. And the reason why you do that is that you can watch when that air bubble enters the pressure myrograph chamber. That's when the solution change occurs, and so you can put a mark on your data acquisition system so that you know that you've achieved solution change. So what happens when we expose the vessel to high extracellular potassium is that we collapse the potassium gradient. So we've exposed the vessel to high extracellular potassium, and we collapse the potassium gradient. So instead of having big potassium currents coming out of the smooth muscle cells, we have small, we have a reduction in the magnitude of the potassium current. So that results in depolarization of the smooth muscle membrane potential, and that activates voltage-gated calcium channels. So we get calcium influx, and that causes vasoconstriction. And so this is what it might look like on your monitor screen. So you see we have a nice big fat vessel here, and then when we expose to the high potassium PSS, we can see we get this massive vasoconstriction. So this should be the first thing you do with your blood vessels, is that you should expose them to high potassium PSS. And if you don't get this big vasoconstriction effect, then something's gone wrong. I mean, this is a real hammer. If your vessels don't constrict to these high concentrations of potassium chloride, then something's gone wrong. Either the tissue has been not well maintained, or your pH is off, or something like that. So this is a good first step, but hopefully you won't encounter any problems. So the second example experiment that I want to talk about is phenylephrine-induced concentration. And so this would be a phenylephrine concentration response curve. And so we know that phenylephrine is a neurotransmitter that binds to the type 1 adrenergic receptor, or alpha-1 receptor, on smooth muscle cells. And so basically through a number of pathways, this causes an increase in intracellular calcium, and this causes smooth muscle cell contraction and vasoconstriction. So what we're going to do is I'm going to show you some examples of what happens when we expose an isolated vessel to increasing concentrations of phenylephrine. So in these examples, we've started at 10 to the 9th molar phenylephrine. Again, this is administered in the superfusate. And then we've increased the concentration in a stepwise fashion from 10 to the -9 all the way up to 10 to the -5 molar phenylephrine. And so you can see we get this, this kind of graded constriction of the blood vessel. Not much going on here at 10 to the -9, but we're starting to get quite a good constriction when we get to 10 to the -7, and it seems to be getting maximal somewhere between 10 to the -6 and 10 to the -5 molar phenylephrine. So this is a real good, if you can see this kind of response, this is a real good way to know that your blood vessels are happy, that you've achieved the proper conditions within your recording chamber. Just to show you what it might look like on your data acquisition system, this shows you a recording of vessel diameter, shown on this axis, as a function of time as we've administered increasing concentrations of phenylephrine. So again, at 10 to the -9, not much going on. We get a little constriction at 10 to the minus 8, a better constriction at 10 to the minus 7, and then much bigger constrictions at 10 to the minus 6 and 10 to the minus 5. One thing I want to point out to you here, this recording is quite instructive in that it shows that what's happened here is that we've lost contrast. So because the vessel is constricted so much, we've had a problem maintaining contrast and maintaining the tracking of the diameter of the vessel. But what you can do is you can adjust the level and the position of the tracking bars, and if you do that, you can actually recover, and you can actually kind of save this recording, and you get a nice stable recording here. So the investigator here had done that. They'd lost their tracking, so they manipulated the level and the position of their recording of the detection bars until they actually were able to obtain a stable diameter. So that's really important. So that's one thing that you need to learn to do as an investigator is basically how to maintain tracking in the face of some of these phenylephrine, that you allow the vessel to achieve a steady state diameter before you administer the second concentration. So you can see here that we've got these nice steady state responses. So this is what you want to measure. You want to measure these steady state responses. You don't want to, you don't want to worry about what's going on here with some of these transient things. What you want to focus on are these steady state responses. So as a reviewer, I review, when I'm reviewing manuscripts, I always ask authors to provide me with example traces so I can make sure that they're actually looking at steady state responses. Okay, so after you do this, after you do a few vessels, you get these diameters, you get these measurements. You want to plot the data, and so this is an example, and these are just kind of made up data. We didn't actually do this experiment, but I sort of wanted to show you what you might want to, what your data might look like. And so what we plot is we call it percent constriction, and this gives you a formula for calculating that, and we'll plot percent constriction on a log plot, on a log scale plot, as a function of phenylephrine-induced concentration. So what you want to do is do 4 or 5 vessels, and then plot this, and your data should look something like this. You should get a nice concentration response curve, and and if you can achieve this, then you have some confidence that you know how to do these experiments and that your system is working appropriately. So the third example I want to talk about is acetylcholine-induced relaxation. So this is a way of looking at endothelium viability. So this is a way of determining whether or not that through your isolation procedures that the endothelium in your vessel is working appropriately. And this just kind of shows you schematically what we're talking about. We know acetylcholine will bind to muscarinic receptors on the endothelial cell plasma membrane, and then through some intracellular signaling pathways, that will cause the production of both nitric oxide and endothelium-derived hyperpolarizing factor. These two pathways will act on the underlying smooth muscle cells, ultimately causing a decrease in intracellular calcium in the smooth muscle cells, and this will cause relaxation of the smooth muscle cells and vasodilation. And this has become the gold standard for assessing intact endothelial function. So if you can get in a mesenteric artery, this is vascular bed dependent. Some vascular beds it doesn't work as well, but in mesenteric arteries you should be able to see a robust acetylcholine-induced vasodilation if the endothelium in your blood vessels is intact. So, and this is what we're talking about. So this is how you would do the experiment. And so we're measuring baseline diameter here. So we need to give the we need to have some initial tone on the blood vessel, and so that when we administer the vasodilator agent, it has something to work against. And so in this case, we're going to pre-constrict the vessels. We're using 1 micromolar phenylephrine. So we've superfused the vessel with 1 micromolar phenylephrine. We've obtained a steady state response. So the vessel is nicely constricted. And then in this case, we've co-administered 10 to the minus 4th molar acetylcholine and 1 micromolar phenylephrine. So you can see that the acetylcholine caused the blood vessel to dilate. To show you what this might look like on your data acquisition system, again, we have the steady state diameter here after administering phenylephrine. So this is our pre-constriction. Then we administer the acetylcholine, and you see you get this very nice vasodilatory response. So then to take this a step further is that you can damage the endothelium. And oftentimes when you're thinking about endothelial function, you want to first assess a response with an intact endothelium and then see how damaging the endothelium impacts that response. And so the way we damage the endothelium in my lab is that we perfuse the vessels with air. So air seems to disrupt the function of the endothelium. So we introduce about 2 mL of air into the perfusion of the blood vessel. So this is what the blood vessel might look like under normal conditions. This is what it looks like when you're perfusing the vessel with air. So you can see that. And so then we repeat the experiment that we just talked about. So we have our baseline diameter. Again, we pre-constrict with phenylephrine, and then we add acetylcholine and phenylephrine together. And so you can see here that we're still seeing a little bit of vasodilation in this blood vessel. We haven't completely removed the effects of acetylcholine, but they're considerably blunted compared to the effects that we got with the intact endothelium. So then again, you go ahead and you plot the data. So we show percent vasodilation, and essentially this is the percent reversal of the pre-constriction. This gives you a formula for calculating this if you want. And this is what the data might look like. So if you do, say, 4 or 5 vessels, so you might want to do these with the endothelium intact, which is shown plotted here. So this is percent vasodilation, again on a log scale as a function of the acetylcholine concentration. So you get a nice concentration response curve. And then you could plot this in vessels in which the endothelium has been damaged, and you should see a significant rightward shift of this curve. So you should see much less vasodilation. So if you can do all of these, then you're in pretty good shape. In terms of using this technique in order to investigate endothelial function. So the fourth example that I want to talk about, and I'll just talk about this real briefly, but this is how you might assess the myogenic response. And so the example I'm going to show you are in cerebral parenchymal arterioles. And so again, these are very small blood vessels, and so this shows you a parenchymal arteriole that we were studying in the lab. You can see how we've tied off this end, but on this end, rather than cannulate the blood vessel, we merely tied it onto the cannula. That's just because these vessels are so tiny and so delicate to work with that you want to minimize how much you handle them. And as long as you can seal the vessel off, it's okay to do something like this. So this vessel is 35 microns in diameter. So this is close to the limit, I think, of what you could study using this technique. So if you want to assess the myogenic response, what you simply do is, this is much easier than the experiments we were talking about earlier, is that you superfuse the vessel with PSS, and then you apply these discrete pressure steps. And we do this using the servo control system. And so in this example, we started the vessel off at a very low level of intraluminal pressure, 5 millimeters of mercury, and then we upped it to 20, 40, 60, 80, 100, 120, and 140 millimeters of mercury. And so you hold the vessel at each pressure until you see basically a steady state diameter. And then you go onto this vessel at about 35 millimeters of mercury, and in spite of this increase in intraluminal pressure, this vessel is actually maintaining its diameter quite well. So it's generating this distending pressure. So then after you've recorded the active responses, then you superfuse the vessel with the calcium-free PSS. And I usually do this for 15 or 20 minutes. And then I start the pressure response curve again. I turn the pressure back down to say 5 millimeters of mercury, and then I do my stepwise increases in pressure. And what you can see here is that this vessel is just dilating passively. The muscle is generating very little, if any muscular tone, and so you can see that the diameter increases in accordance with the increase in intraluminal pressure. So to plot these data, we calculate myogenic tone, and it's basically the difference between the passive and the active diameter as a function of intraluminal pressure, and then you might see a response curve like this. Okay, so now we've been through 4 example experiments. So just in terms of some tips and pointers, the big thing, if you're new to this technique, the big thing that I can recommend is that you practice. So some people, if you have very good manual dexterity, you're really good at doing this, you might learn to do this in a week or 2. If you're not, you might take 2 or 3 months. Most people take about a month to get really good at this, is my experience in my lab. And so what I have the people in my lab do is I do the procedures in experiments 1 through 3. So potassium chloride-induced constriction, phenylephrine concentration response curve, ACH-induced dilation. And we do this usually using 2nd or 3rd order mesenteric arteries that are on the order of about 200 microns in diameter. So you do this until you get consistent results. And so I have a new postdoc in the lab, and that's actually what he's doing today. He's doing phenylephrine concentration response curve until it gets good at it. So just do this using mesenteric arteries, and then once you're confident, once this is working in your hands, then move on to your vessel of choice, whatever you feel comfortable working with. The other tip, work fast. You want to remove the organs. So after you euthanize the animal, try to get the organs out and isolate the vessels as quickly as possible. My experience is on cerebral arteries, this is really critical. If you leave the blood vessels on the brain too long, toxins released from the neurons as they die seem to poison the blood vessels, and it seems to reduce reactivity. So if you want to get consistent results, work fast, get the organs out, get the vessels off as quickly as you can. Again, make your solutions fresh each day. Don't get lazy. Don't try to store them overnight. You'll be sorry. One thing we didn't talk about is, and this is something that's really hard to talk about, is one of those things that you really need to see, but is stretching the vessel in the initial prep. So you want to apply physiological levels of stretch to the blood vessel, but how do you do that? And the way we do that in my lab is that you pressurize the blood vessel. So you pressurize it, say, to maybe 40 millimeters of mercury. You look at it under the microscope, and what will happen is the vessel should bend a little bit. So it should, instead of being straight, it'll have more of a C shape. And so what we do is that we just move the cannula apart until we remove that bend, and then maybe you just give it just a hair more stretch after that, just a tiny bit more. And that seems to give you consistent results. But that's one of the things that you really need to learn. You need to see this. You need to do it on a daily basis. And then you'll get to the point where you know exactly where you need to be in order to get reproducible results. It's important if you're having problems, the first thing to do is to check your pH, your oxygenation, your temperature. And so you can get electrodes to measure this in your bath. So just make sure you're getting consistent conditions in your bath. The other thing that I would say is that this is a technique that it's better if you do it several days a week rather than just do it like 1 day a week here and there. Just schedule some time and plan on doing it 2, 3, 4 days a week until you get good at it. You'll learn much faster that way. It's quite frustrating if you just can kind of move in once in a while. So try to set aside a good block of time if you want to learn this technique. And so then I've gone over my time, so I'm just going to wrap it up, and I think we'll take some questions. And thank you very much for your patience and for listening to me today.
Thanks, Scott. That was a great overview, and yeah, we've had a bunch of responses from the audience, many of which have said, just the content in your section has perhaps answered some of their questions that they had coming into this event already. So that's great. And yeah, we are running a little bit late, but again, there's an opportunity here for a short Q&A, so we're going to move forward with just a few questions, taking into consideration the time that we ask people to block off. But so if you do need to rush on to something else, just remember this is being recorded. You will have access to it later. And all of the questions that have come in, because we will not get to all of them, will be answered by our presenters and then prepared in a summary report, which you can download later. So I'll just check if we also have Gerry on the line. Gerry, are you there?
Yeah, I'm here. Thanks, Andy.
Perfect,
Thanks,
perfect. All right, so yeah, let me fire the first question at you guys. So we've had a few questions come in about basically microscope, what objectives should be used, how best to visualize the artery, and also whether an upright microscope can be used. So can you guys clarify that?
Yeah, I can start with a little bit of perspective. Most people find it, regarding the microscope, an upright microscope can be used, but it's generally preferable to use an inverted microscope because a lot of times you're going to want to have some additional accessories, like maybe a pH meter or electrode if you're doing intracellular membrane potential recordings, oxygen electrodes. You might have some accessories that you want to have in the bath, and so you want to have that extra room above the chamber to be working with, and so most people find it's preferable to use an inverted scope for that reason. So as far as objectives and powers, 10x is a good objective lens to have. Most of the microscopes available will come with a range of objective lenses, but 10x is a good one to have. For smaller vessels, 20x. Maybe for the stuff Scott's doing, you'd want to go up to a 40x with those cerebral arterioles at 30 microns, but you know, generally a 10x or a 20x is probably where most people are going to be at. So.
Yeah, I would agree with Gerry on that. Our scopes are equipped with 10 and 20x, and you know, every once in a while we'll go to a 40 for the really challenging experiments. And we've never used an inverted microscope, or I'm sorry, an upright microscope. All of our scopes are inverted.
Okay, perfect. Thanks, guys. Another one where we've had a variety of different questions come in, maybe with specific points, which we'll address in the Q&A summary. But in general, can you guys speak to the kind of high and low range as far as what size blood vessels you can work with in a pressure arteriograph?
Yeah, again, we certainly hear down to, as Scott mentioned, 30, 35 microns, 40 microns. That's pushing the lower limit of what is generally possible, all the way up to 300 microns. I would say most applications are probably between 1 and 200 microns for the blood vessel size. Certainly based on the diameter of the blood vessel, that's going to dictate the cannula diameter that you're working with. So you just want to make sure you have appropriately sized cannula to match the blood vessel diameter that you're working with.
I would agree with Gerry on that.
Okay, perfect. All right, and then I guess on the low end, or a question relating to very small, working with very small vessels. Someone has asked, are there any tricks to cannulate brain vessels such as the middle cerebral artery or basilar? They're finding that they always have side branches and it's very difficult for them to work with this particular vessel. Do you have any specific suggestions?
Yeah, well, I mean, all I can say is, yeah, I agree, because it is. I mean, it is challenging, and that is one of the things. We don't use middle cerebral arteries very often for that reason. That they tend to be branchier. So we'll use the cerebellar arteries, the superior cerebellar artery, and then we'll use the posterior. It's called the third branch of the posterior cerebral artery. So we tend to be able to isolate segments that are of sufficient length to allow cannulation without side branches. Without that, there are some. If you go to the very distal portion of the middle cerebral artery, sometimes you can find segments that are long enough to cannulate. But yeah, that is very challenging, and I mean that's something that we go through every day. But it's possible. You just need to be patient and look for the appropriate vessel segments.
Yeah.
Okay.
I would just add to with the branchy vessels, if you really, if that's the vessel you need to study, it is possible to, although tedious, to isolate branches with ligatures. It does require a lot of extra effort in the mounting process, but some people do take the extra time to ligate side branches on those branchy vessels.
Okay. All right, very good. There were also a number of questions that talked about, specific techniques during the isolation phase of a vessel, and then how long it can be stored in a buffer. Scott, you mentioned you guys store these, and perhaps you can provide a little bit more detail about the type of buffer, maybe the temperature, the composition, so that these samples stay intact and viable for your experiments moving forward.
Yeah, I mean, we use this MOPS buffered saline solution, and again, if you can look at my papers and get the composition, but this is a solution that contains. We put some albumin in the solution to help stabilize the vessels. It's got glucose and pyruvate and things like that. It's pH'd. It's a biological buffer, so it's pH'd at 7.4, so you don't have to bubble it, which is convenient. And then we'll isolate a set of vessels and we'll store them on ice in that solution, and probably maybe 6 to 8 hours. Usually that's a typical experimental day. I wouldn't go too much longer than that. I mean, some people talk about holding them overnight and things like that, but I would worry about loss of viability. So typically, the workflow is you come in the morning, make the solutions, isolate the vessels, and just store a set of vessels on ice for, say, 6 or 8 hours, and that's usually enough to last you through the day.
Okay. Very good. All right, guys. Well, in the interest of time, we have a ton of questions that have come in. So for all the audience that's still with us, thank you for submitting questions, for staying, the extra length during today's webinar. But I'm going to suggest that we close things off. And again, just for everyone that's online, all the questions that have come in, these are going to be reviewed by our presenters, and answers will be put together in a summary report. The slides from today, as well as an appendix on some of the buffer recipes that were discussed, they're all going to be available for download, as well as the full recording. So at this point, yeah, I'd like to officially thank Living Systems Instrumentation, Doctors Gerry Herrera and Scott Earley for your contributions today. This was an excellent event, and our audience has confirmed that with all the feedback. So many thanks to you, and we hope to have everybody back on a future event soon.